Now coming to this non-classical inheritance, in which you find that there's trinucleotide repeat mutations, mitochondrial genes mutations, genomic imprinting, and gonadal mosaicism. If you talk about trinucleotide repeat mutations, obviously I have told you the basis before that there is expansion of the trinucleotide repeats, okay, and it is also very important in the neurodegenerative disorders, and is, as I've already mentioned before, that these two nucleotides are almost always present. So what are the mechanisms happening? Number one, if you talk about the loss of function in the gene, that will affect the transcription. There's going to be transcription silencing, for example, in the Fragile X syndrome, which is happening in the non-coding part of the gene. Next mechanism is the gain of function mutation because of alteration of the protein structure happening in the Huntington disease and the spinocerebellar ataxia. And that is happening in the coding regions. Third point is the gain of function, which is mediated by the RNAs, which is found in the Fragile X syndrome, and that is happening in the non-coding parts. So these, sometimes you may get questions, you may get MCQs confused. You must remember these three mechanisms. But what is more important and most important is this table. Very, very important. You must know that there is something called as non-coding regions and coding regions. The repetitions or the expansions which are involving the non-coding regions, this happens during oogenesis, and the expansions affecting the coding regions is happening during spermatogenesis. Now, these diseases, the genes, the repeats, and the proteins and the locus, all of this is very, very important. You may not remember this, okay, you may not remember this, but this portion is very, very important. But it is very simple also. See, locus, now you don't have to remember the entire thing. What you can do is that you can just remember the chromosome. Fragile X syndrome, the chromosome affected is X chromosome. Myotonic dystrophy, it is the 19 number chromosome. Spinobulbar muscular atrophy or Kennedy disease, it is the X chromosome. Huntington disease, 4 number chromosome. Hoare syndrome, 12. Spinocerebellar ataxia, it is type 1 is 6, type 2 is 12, and type 3 or Mercado-Joseph's disease is chromosome number 14, type 6 is chromosome number 19, and 7 is chromosome number 3. Again, this is a very volatile chart, but very, very important. The proteins involved are so important. In Fragile X syndrome, it is the FMR1 protein involved. Myotonic dystrophy makes the myotonic dystrophy protein kinase involved. In Kennedy disease, it is the androgen receptor which is involved. In all this, in the Hoare syndrome, you have the atrophin 1 which is involved. In all the ataxia diseases, you have the ataxin which is involved, except the type 6 where you have the voltage gated calcium channel subunit, which is the protein involved. Very, very important. All these points are so important. But the repeats are very easy to remember, and they are must remember. Most important is the repeats. All this is important. In the coding regions, all these diseases is affecting this CAG codon. CAG codon. In the Fragile X syndrome, you have the CGG repeat. In the Friedreich's ataxia, you have the GAA repeat, and in the myotonic dystrophy, you have the CTG repeats. Only this part is difficult. You have to remember this. Again, I would ask you to write them down or take a screenshot and keep reading them and repeating them. These genes are also important, but most important is the repeats and the proteins. Very, very important. Another important point I would want to mention here is about the intranuclear inclusions. So, a morphologic hallmark of these diseases is the accumulation of aggregated mutant proteins. So, I've already explained to you how these proteins are getting degraded. I've already explained you about the ER stress response, right? The protein stress response. So here there is accumulation of the mutant proteins. The body is no more able to combat it. There is accumulation of these proteins, and therefore there is a large intranuclear inclusions. This is also an important point, okay, in the trinucleotide repeat disorders. So now we're going to read about each of these disorders in detail, starting with the Fragile X syndrome, which is very, very important. Okay, so this is a most common genetic disease of intellectual disability in the males, and the second most common cause after Down syndrome. Very, very important. These patients, these children, they do have intellectual disability. It results from a trinucleotide expansion mutation in the familial mental retardation 1 or the FMR1 gene. Very important. Now there are two other disorders having the same mutation, that is the Fragile X associated tremor or ataxia syndrome and Fragile X associated primary ovarian insufficiency. Now you must understand something. Before I explain you this, I want to tell you something, that what happens is that there is something called as pre-mutations and there is something called as full-blown mutation. So what happens is, as I've mentioned here, that Fragile X syndrome, it is happening in the non-coding regions, and in the non-coding regions it is happening during oogenesis, right? So what usually happens is that during oogenesis, when the females are carriers, the females are usually carriers, and during the oogenesis, when there is production of the progeny, there's dramatic amplification of the CGG repeat, right? So which means that with every successive generation, with every successive progeny, the amplification of this trinucleotide repeats keep increasing. Okay, and once there is dramatic amplification of this CGG repeats, now this pre-mutations is getting converted to full-blown mutation, and now there is ultimately occurrence of this Fragile X syndrome. So mental retardation is happening in most male child, but it's only happening in 50% of the female child. So see, why it is called as Fragile X syndrome? Because the cytogenetic abnormality or the alteration what is happening in the X chromosome of the FMR1 gene, what's happening is it is being shown as a discontinuity of staining or as a constriction in the long arm of the X chromosome. When it is being cultured, it is shown that there is some kind of discontinuity in the staining which appears as broken. It seems as if it is broken at that particular point in the long arm of X chromosome. Important point. That is why it is named as Fragile X syndrome. The males with Fragile X syndrome, they have marked intellectual disability. The patients have a long face with large mandible, large everted ears, large testicles, hyperextensible joint, and high arched palate. Mitral valve prolapse noted in some of the patients. They mimic a connective tissue disorder. The most distinctive features is macroorchidism. Very important point. In addition to intellectual disability, there's several other neurologic and neuropsychiatric manifestations. They can be epilepsy, autism spectrum disorders, all of which can happen here. Okay. Now, if you talk about the the carrier males will transmit through their phenotypically normal daughters to the affected grandchildren. So they are normal transmitting male. Okay, they are normal, but they're actually transmitting their disease to the affected grandchildren, and therefore I said that with every passing generation, the grandchildren are affected more than the children, right? Now, what is anticipation? Very important point. Fragile X syndrome shows this feature of anticipation. Very important MCQ. What is the meaning? The clinical features of this worsen with every successive generation, as if the mutation becomes increasingly deleterious as it is transmitted from a man to his grandson and great grandson through the daughters. So you understand, with every successive generation, these mutations are amplifying and the effects are becoming more serious. And this is what is called as anticipation seen in Fragile X syndrome. So what is pre-mutations? Initially, in the initial generations, in the initial progeny, it is only 55 to 100, right? But then with every successive generation, there is full-blown mutation reaching up to 4,000 repeats. The carrier males transmit the repeats to their progeny with small changes, but when this mutation is in the carrier female, because this process is happening in the oogenesis stage, therefore if the male is a carrier, there is not much amplification. But because this is happening in the oogenesis phase, therefore if the carrier is a female, then with successive generation you find that there is dramatic amplification and there is intellectual disability in the male offspring and 50% of the female offspring. Thus, it seems that during the process of oogenesis, but not spermatogenesis, pre-mutations can be converted to mutations by triplet repeat amplification. A very important point. During oogenesis, the pre-mutations is getting converted to full-blown mutations by the process of triplet repeat amplification. Very, very important point. The likelihood of intellectual disability is much higher in the grandson than in the brothers. I've already mentioned this before. Now, this protein, which is the Fragile X mental retardation protein, or the FMRP, this is the protein which is produced by this gene, and this protein, it selectively binds to mRNA, and it will help in this intracellular transport of these polysomes to the dendrites. It is basically a translation regulator. So whenever there is a reduction in the FMRP in this particular syndrome, that will result in an increased translation, right? And because of this increased translation, there's an increase in production of the proteins at the synapses, which result in loss of synaptic plasticity, the ability of the synapses to change and adapt to response to specific signals. So basically, because this protein is having a role in kind of transporting the polysome, and it is also a translation regulator. Now when this protein is defective, this translation is increasing, and because of which the proteins are also increasing, and because of this increase in protein, there is a loss of synaptic plasticity. Plasticity means that the synapses have the ability to change and adapt according to the response. But once there is a loss of this plasticity, therefore this patient is having so much of mental disability. This learning and memory is so much affected because of this mechanism. Now, since I mentioned about the pre-mutations, I've already told you what is the pre-mutations, right? I've explained you here what is the pre-mutations. Now you must understand that pre-mutations के time पे क्या होता है? Pre-mutations के time पे the other two syndromes that we have learned about, the Fragile X associated primary ovarian failure and Fragile X associated tremor or ataxia. These are the disorders that happen when the pre-mutations was going on, and once the pre-mutations get converted to full-blown mutation, then the Fragile X syndrome actually happens. So you can see here that if you talk about the Fragile X syndrome, it is because of transcriptional silencing of the CGG repeat, and when there's transcriptional dysregulation, there's accumulation of toxic mRNA happening, which is ultimately causing these two syndromes. And mind you, this two syndromes is happening when the pre-mutation process is going on, and once the pre-mutations get converted to full-blown mutations, then you will find this Fragile X syndrome happening. Okay, this is GAA triplet, same process, Friedreich's ataxia, CAG triplet, same process, Huntington's disease. This is what is mentioned over here, that during the process of pre-mutation, as I've shown you over here, that initially there's pre-mutations, and with every successive generation there's mutations. So during the initial stages of amplification, which means when the amplification is not that huge to cause Fragile X syndrome, then during those pre-mutation stage, the other two syndromes, that is this syndrome and this syndrome, is happening. Okay. Now coming to the mitochondrial genes, obviously the Leber hereditary optic neuropathy is an example of this, very, very important. You must remember these examples. Leber hereditary optic neuropathy is a prototype of the mitochondrial disorder, very, very important. There is a visual impairment noted, and there is bilateral progressive loss of central vision.